So we did the branch diagram. That is where we had stopped the earlier day, and we had done this example. Sorry, where did that example go? We had done this example the other day, where I had said you that when we do this kind of crosses, and if there are two characters, as I said, this is dihybrid, so there are two characters or two different genes are in consideration. We have to calculate the individual probability of inheritance of each of the gene, then multiply it to get the combined probability. So this is what we did the earlier days. I'm not going over this. For a recapitulation, I'm going over this cross. So you see there is a round yellow seed and another round yellow seed. Now if we look at the genotype of the two genes in concern, so capital R small r into capital R small r, that should be considered separately, and capital Y small y into capital Y small y, that should be considered separately. If we remember the traditional Mendelian monohybrid cross ratio, when we cross a capital R small r with a capital R small r, the ratio of the capital R phenotype is 3 and the small r phenotype is 1. So it is 3:1. You have done this Mendelian monohybrid ratio in your 11-12. Okay. So from this individual cross, we can say that the probability of having round seeds is 3/4 and the probability of having wrinkled seed is 1/4. Now we calculate the individual probability of the cross between a yellow seed with another yellow seed. So this is a heterozygous cross of a heterozygous yellow seed with another heterozygous yellow seed. So capital Y small y into capital Y small y. Now if we think of the gamete composition and then the products of the cross, again we get 3/4 yellow and 1/4 green. Now as I said, when we are considering a dihybrid cross, we multiply the individual probabilities to get the overall probability for the full cross. So what we do? 3/4 round plant is once multiplied with 3/4 yellow seeds and once multiplied with 1/4. So we get 9/16 round yellow. Okay. So and we get 3/16 simple multiplication round green. And then we have this probability of wrinkled 1/4. 1/4 multiplied with 3/4 wrinkles in multiplied with 3/4 yellow, that is 3/16, and 1/4 wrinkles in multiplied with 1/4 green, that is wrinkled green. So this is how actually when crosses are done. I'll be showing you an example of a trihybrid cross, but I'll come to that later on. But let me just get into the basic conclusions that Mendel came from his two crosses. As we discussed other day, from the monohybrid cross, Mendel proposed that each plant must possess two genetic factors encoding a character. And here the two genetic factors encoding a character is capital R small r. So let's look at the monohybrid cross. Now, so this is the dihybrid. This was a monohybrid. So each plant must possess two genetic factors encoding a character. So the round character was encoded by these two alleles, capital R, capital R. The second conclusion that Mendel drew from this experiment was that two alleles in each plant separate when the gametes are formed. So the two alleles separate when the gametes are formed and one allele goes into each gamete. Now the most important part of the conclusion from this cross was the concept of dominance. He proposed that the traits that remain unchanged in the F1 heterozygous offspring. So this is a F1 heterozygous offspring. So the traits that remain unchanged in the F1 heterozygous offspring, Mendel called them dominant, and the traits those disappeared in the F1 heterozygous offspring, he called them recessive. When dominant and recessive alleles are present together, the recessive allele is masked or suppressed. The concept of dominance was another important point that came up from Mendel's monohybrid cross. So as you can see, if this is the heterozygous plant having round seeds, the two characters in consideration was the round seed and the wrinkled seed. See, this is phenotypically they are different. But when we look into the heterozygous seed, we are seeing that the character is that of this parent only. There is no phenotypic expression of these character in this particular F1 generation. That means the round character is completely masking the wrinkled character. So round is dominant over wrinkled, and thus capital R represents the dominant allele and the small r represents the recessive allele. So this was some very vital considerations that came up from Mendel's monohybrid experiment. When we go by the dihybrid experiment, as we started with that today, the two vital, the main vital theory that came up from this experiment was the principle of independent assortment that tells us that the alleles at each locus separate independently. So capital R and small r are separating in an independent manner and segregating in a definite pattern, and capital Y, small y are also segregating in a definite pattern. And when they are inherited, there is no interdependence between the capital R allele and or the small r allele with the capital Y allele or the small y allele. So this is where his hypothesis of independent assortment stands, and the concept of dominance and recessive also comes up. Now let me go to a trihybrid experiment, but before coach, okay? Now coming to a basic trihybrid cross. Trihybrid cross is not in your syllabus, but just to show you that how by simply calculating the individual properties, you can easily deduce what would be the final progeny. This is simple an example to show you how individual probabilities can be calculated and then they can be combined to give us a final proportion. So let us see. So this is a trihybrid gamete. So capital A, capital B, capital C. And we have small a, small b, small c. Gametes gets separated. And in the F1 there is cross. When you cross this gametes, you will get a combination like this. So we have a capital A, small a, capital B, small b, and capital C, small c. And these are the all possible gametes that can be obtained from this. But to ease our workflow, let us assume what would be the proportions. Now calculating the proportions becomes a bit difficult when we deal with multiple alleles. Now I wouldn't say multiple alleles, when we deal with multiple genes. So let us do it the simple way. Now if we cross a capital A, small a, capital B, small b, capital C, small c with another same genotype plant, so let us now look at the individual probability. When a capital A, small a is crossed with a capital A, small a, it's a monohybrid cross. Take it individually. What do we get? 3/4 capital A phenotype, 1/4 small a phenotype. So this is it. We get 3/4 capital A phenotype, homozygous plus heterozygotes, and we get 1/4 small a phenotype, the homozygotes. So when we cross a capital A with a small a with a capital A small a, we get 1 capital A, 2 capital A small a, 1 small a small a. So the total capital A observed is 3 parts, 3, and the small a observed is 1/4. Now when we do the same, considering this gene differently. Capital B small b into capital B small b. Think of it as separate. Now again we get 3/4, that is 3 parts capital B phenotype, 1 part small b phenotype. So 3/4 capital B plus capital B small b and 1/4 small b small b. So that is how. So the first 3/4 capital A, we now multiply both of these factors, and with the 1/4 small a we multiply both of the B probabilities of the B allele. Okay. Now there is a third allele. Now third allele also will be taken separately. Capital C small c into capital C small c will give us again 3/4 capital C phenotype, 1/4 small c phenotype. So we multiply the probability of A and B now combined with the probability of C individually. So we multiply B with C/4, 3/4 capital C, 1/4 capital C. We multiply B with small b with 3/4 capital C, 1/4 small c. Similarly, for the small a allele, we multiply capital B with 3/4 small capital C, 1/4 small c, and we multiply the small b with 3/4 capital C, 1/4 small c. If there had been a fourth allele, the process goes in the similar manner. Then we would have multiplied this capital C with 3/4 capital D and 1/4 small d. This small c with again 3/4 capital D, 1/4 small c. So whatever. So this is how when we multiply, we are combining the probabilities of now the three alleles. So let us look at the first case. 3/4 into 3/4 into 3/4, 27/64, capital A, capital B, capital C. So this is how, instead of going through the individual crosses in a checkerboard pattern, it becomes difficult when you deal with more than 2 characters. Then it becomes very difficult to calculate that phenotypic expression or genotypic expression in this checkerboard pattern. So this is the traditional, this is the mathematical way how then the probabilities of how many plants that will come up and in what ratio proportions they will be obtained is obtained by this process. So we got the basic concept of how to do a trihybrid cross and the basic concept of dominance and recessive from the Mendelian experiments. Mendel clearly pointed out that if a trait is marked in heterozygous condition, that is recessive, and the one that expresses is the dominant. Now let us look into some deviations from the traditional Mendelian experiments which Mendel had observed. And now comes the question of incomplete dominance and codominance. When we think of incomplete dominance, we have to imagine that one allele of a gene is not completely dominant to another allele of the same gene. It is said to show incomplete dominance, also at times referred to as semi-dominance or partial dominance. Now let's look into this picture and let's compare the situation. This is an example of a complete dominance, and you can see this is A1 which encodes the red color. So this is a gene A1, and this is A2 which encodes a white color. We don't know which one is dominant and which one is recessive. Let us suppose that we do not know which one is recessive and which one is dominant, and hence both the symbols are given the same importance. None of them are written in small. Now A1 encodes red flowers, A2 encodes white flowers. Okay, pure breed, homozygous. Now when a cross between two is made, if the If the F1 generation of the heterozygotes are observed as red colored, then we will presume without doubt that A1 is dominant to A2. But if the F1 genotype of this cross is observed to be a white flower, then we have to presume that A2 is completely dominant over A1. So you can see that either the red color can completely mask the white color. If the red color completely masks the white color, then red is dominant. If the white color completely marks the red color, then the white is dominant. So it is the condition of the heterozygous which actually tells us which character will be dominant and which character will be recessive. So for Mendel, when it was a round seed was crossed with wrinkled seed, and in the F1 heterozygous, through the experiments we saw, all the seeds were round. So obviously round was dominant to wrinkled. Same with the height of that plant experiment, where we cross a tall plant with a purebred dwarf plant. And the F1 heterozygotes were observed to be tall. So tall was dominant to the dwarf, and the tall completely masked the dwarf character. Now let us come to this condition. As I said, incomplete dominance arises when the gene is not completely dominant to another allele. So if red is one character, one parent, and if white be the other parent, so we have two extremes. And let us suppose for this experiment, for this particular example, let us suppose that A1 is supposed to be dominant to A2. Let us suppose. Or let us assume that we do not know who is the dominant ratio. Now, if we have a red parent and we have a white parent, and the cross is done, and we see that the F1 heterozygotes show a range of characters like this. That means the F1 heterozygote is not showing a red character, but a variation of red character in such a manner that tends to become white. You can see this is tending to become white. This is expressing the most nearer to the red parent, and this is the most nearer to the white parent. So it is showing a range of phenotypic characters. That is, this proves that the red character is not completely dominant over the white. The red is showing dominance. Obviously, the red is showing dominance. As you can see, we are getting a range. But the white character, parts of the white character are being also expressed. So the red is not able to completely mask the effect of the white. And hence, this case, we call this as incomplete dominance. So the A1 allele is incompletely dominant to the A2 allele, as the A1 allele fails to mask the expression, fails to mask completely the expression of the A2 allele. This is an example which is frequently observed in case of real vegetables. This is eggplant or brinjal. So this is the purple fruit and this is the white fruit. And you can see the F1 generation. It's not the this parent. Neither is this parent. It's an intermediate between these two parents with a tendency of having a more affinity towards this parent. So the capital P is incompletely dominant to the small p, and when these F1 heterozygotes are self-fertilized, we see that we get a purple homozygote, the original parent, and a white homozygote, again the original parent. But there are two heterozygotes which are phenotypically different from either of the parent. So in this case, the Mendelian monohybrid ratio of 3:1, which is the phenotypic ratio, actually becomes 1:2:1, So the phenotypic ratio has changed, and the phenotypic ratio shows similarity with the genotypic ratio. So this is the concept of incomplete dominance. I'll go over the another small important point regarding this characteristics of dominance. As you can see, I've written the three definition. Dominance can be defined as the phenotype of the heterozygote, same as the phenotype of one of the homozygote plant. Incomplete, where the phenotype of the heterozygote falls within a range between the dominant plant and the recessive plant, and coming to the concept of codominance. I'll come to that in the next slide, but before that I want to show you something here. Now let us look into this picture. And this is a picture where the allele is showing complete dominance, because the color of the heterozygote flower is similar to the color of the homozygote flower. So from this particular experiment, it is evident that capital P is dominant to small p. That is evident. Now let us look into the functional aspect. If I think of this particular flower, there are two capital P, so there are two genes of capital P. If one gene produces 50% of the product, the other gene is also producing 50% of the product, and we are getting a total of 100% product, right? So since there are two genes, we are getting 100% of the product of this gene, and we are getting a phenotype of purple. Okay, fine. Now let's look at the homozygote. There is one capital P and one small p, so there is one gene of the dominant allele. Here we have two genes. Here we have one gene of the dominant allele, and that one gene is only giving us 50% of the product, as compared to the 100% of the product observed when we had two alleles of the gene. Now this 50% of the product is efficient enough to completely dominate the situation and also produce the purple coloration. So a particular gene or a particular allele who in a homozygous condition or a heterozygous condition gives full phenotypic expression of its product is known as, this kind of inheritance is known as, or this kind of gene is known as haplosufficient gene. I'm repeating once more. In this case, two capital P is producing 100% of the product and we are getting a purple color. In this case, only one capital is producing a 50% of the product and we are getting a capital and we are getting a purple color. There is no difference between the two purple colors. So one capital P is enough to produce sufficient protein which is equivalent to the expression that was produced by two capital P. So here the capital P is known as a haplosufficient gene.